What are genetic markers in primate taxonomy studies? Imagine trying to tell two very similar twins apart. Sometimes, their looks are almost identical, but their DNA can reveal who is who. That's what genetic markers do in primate studies. They are specific parts of DNA that help scientists identify and differentiate primates, whether they are individual animals, groups, or entire species. These markers are like biological fingerprints that stay consistent enough to tell us apart but also chain enough over time to show evolutionary relationships. In primate taxonomy, which is the science of classifying primates, genetic markers are essential tools. They help researchers figure out how different primate species are related and where they fit on the family tree. For example, some primates look very similar but are actually quite different genetically. Without genetic markers, it would be hard to tell if two populations are separate species or just variations of the same one. This is especially important for conservation efforts because knowing exactly which species are endangered helps protect them properly. These markers are found in both nuclear DNA, which is the DNA in the cell nucleus, and mitochondrial DNA, which is inherited from the mother. Common mitochondrial markers include genes like cytochrome C oxidase subunit I, cytochrome C oxidase subunit Roman II, and NADH dehydrogenase subunit 5. These genes are chosen because they change at a steady rate, making them good for comparing closely related species. Other types of genetic markers include microsatellites and minisatellites, which are repeating sequences of DNA that vary a lot between individuals. These are useful for studying how populations are structured and how they change over time. Using genetic markers, scientists can build family trees called phylogenetic trees. These trees show how different primates are related and when they likely diverged from common ancestors. For example, genetic studies suggest that humans and chimpanzees split from a common ancestor about 6 to 8 million years ago. This kind of information helps us understand how our own species evolved and how we are connected to other primates. In the field, scientists can collect DNA samples from blood, hair, tissue, or even feces. This makes it possible to study wild primates without disturbing them too much. The DNA is then analyzed in laboratories to look for specific genetic markers. This analysis can tell us about parentage, how related different individuals are, and how genes flow between groups. All of this information helps us understand primate behavior, social structures, and how they adapt to their environments. Overall, genetic markers are vital for classifying primates accurately, understanding their evolutionary history, and supporting efforts to protect endangered species. They also help us see how humans fit into the larger primate family and trace our own evolutionary journey.